So today we will begin the chapter on vestibular system. As you have already learned on audition, you know the structure and the receptors for audition are situated in the internal ear. But now this is part of again an internal ear where we have some receptors which are functioning for the maintenance of posture as well as equilibrium in our body. Whether the person is in motion or the person is static, we maintain the posture and equilibrium. And this is possible only because of these receptors that are present and they together constitute the vestibular apparatus. So today we will begin with the and you know, as part of function we have the cochlea, which is a coiled structure, and it is a structure where there are bony canals as well as membranous canals called as the name of labyrinths. So we have bony labyrinths and membranous labyrinth. This is actually a continuation of the same part of the bony and the membranous labyrinth situated in the internal ear. So this sensory organ is very complex in nature, but at the same time very sensitive. We will come to know of these sensations only when there is some abnormality. Otherwise, we are not at all aware of the sensation because it's an unconscious sensation. We never know how we are maintaining the posture and the equilibrium, whether we are static or we are in motion like running, jumping, walking, whatever it is. So, this apparatus or the sense organ help us in maintaining the posture and equilibrium in motion as well as not in motion, that is when we are static. Coming to the structure, we will just learn about the functional anatomy and the mechanism how it gets stimulated. So this vestibular apparatus actually is seated or housed in the petrous part of the temporal bone where the structure for the audition is also there. As I told, it's all continuous. Right from the oval window, there it goes to the cochlea. So this is also part of the same internal ear, and we have structures mainly of two parts. One is a semicircular canals, and there are other two sac-like structures called as the utricle and the saccule. So we have, if we take the midline and we have two internal ear on either side, we have three semicircular canals which opens into two sac-like structures which are continuous. So this is a utricle and this is a saccule, and these are the three semicircular canals. As I told you, it's a bony labyrinth with a membranous labyrinth where fluid is filled in both these cavities, the bony labyrinth as well as the membranous labyrinth. The membranous labyrinth is lining the bony labyrinth, so there is a fluid between the membranous labyrinth and the bony labyrinth which is called as a perilymph. So we have the bony labyrinth which is filled with perilymph. At the same time, the membranous labyrinth is there which is filled with a fluid called as an endolymph. Both of them are different for some reasons. So the perilymph and the endolymph will be located in such a way that perilymph is situated within the bony labyrinth and the membranous labyrinth, whereas the membranous labyrinth will be filled with endolymph. And endolymph is secreted by the stria vascularis, which I have already learnt along with audition. So the fluid is continuous in all these structures and it is rich in potassium, whereas perilymph is rich in sodium but not with potassium, and it is produced from the extracellular fluid and it is similar to the cerebrospinal fluid. So those are the differences between the perilymph and the endolymph. Now, these three semicircular canals we'll learn in detail, which opens into a sac-like structure commonly, which is called as a utricle, and in turn the utricle will be opening into another sac-like structure which is called the saccule. So we have the utricle and we have the saccule. This saccule will be continuous with the which structure? Cochlea. So now we say there are two divisions functionally for the structures that are seated within the petrous part of the temporal bone. The semicircular canals along with the utricle and the saccule will be concerned with the maintenance of the posture and equilibrium, whereas the cochlear part is concerned with audition. So we have two divisions of the 8th cranial nerve, which is called as a vestibulocochlear nerve. The vestibular division of the 8th nerve will be innervating these structures, and the cochlear division will be innervating the cochlea. So there are two functions for this for this particular cranial nerve. And now we will go into the details of this semicircular canals, where these canals are actually lying in such a way where they are perpendicular to each other. Follow. And these semicircular canals are named as, one is the anterior one, otherwise called as a superior one. Then we have another which is a posterior one, or otherwise it can be also called as the inferior one. And we have another canal which is called as a lateral one, or it can be otherwise called as a horizontal one. So we have the anterior or the superior, posterior or the inferior, and the lateral or the horizontal. So accordingly we have the same structures on the right side also. But there is something different where when they function together, because we have on either side. The vestibular apparatus is present on either side. And these canals will be presenting its position in such a way that if a person inclines the head 30 degrees in the forward direction, they all will be directed in such a way that the anterior canal will be directed outward and anteriorly. Follow. It will be directed anteriorly as well as outward. The posterior canal similarly will be directed outward and posteriorly. And the horizontal or the lateral canal will be lying parallel to the ground. This is how these canals will be directed or oriented when a person inclines the head 30 degrees forward. And we have to understand one more point where this anterior canal on the right side, let us say this is the right side and this is the left side. This anterior canal on the right side will be in the same plane of the posterior canal of the left. So the anterior will be in the same plane as the posterior of the inferior on the left side. Similarly, this posterior, because it is in line in plane with the anterior, similarly we have the same anterior in plane with the posterior of the right side. Is it clear? So they are in the same plane. This is specially mentioned because they always function together, not on the one side. So here again when we take the lateral one or the horizontal one, they will be in the same plane. So this is how it looks like. The anterior of the right in plane with the posterior of the left, the anterior on the left in plane with the posterior of the right. The horizontal or the lateral will be in the same plane. So whenever they function together, we see that they are just like mirror images. Moreover, the action can be described as being pushing and pulling or a push-pull fashion. If one side it is a push, we are considering it as a push, the other side will be a pulling effect. We will come to it later. So this is how these canals will be functioning. So this is just an orientation of the canals. Going to more details of the canal, we see that these three semicircular canals are opening into the utricle, the next sac-like structure. So they have two free ends, which we will consider as 6 openings through which these 3 canals will be opening into the utricle. But there are only 5 openings. This is because the anterior and the posterior together will be sharing a single opening. So these 3 canals will be opening into the utricle by means of 5 openings and not 6 openings. So there are 2 free ends for all the semicircular canals, but one free end of all will be having a dilated structure which is now called as the ampulla. So each canal, the free end is there, one end will be dilated. Again for the horizontal canal we have the free ends, one end which will be dilated. Again for the posterior one we have 2 free ends, one will be dilated. It is this dilated portion which is now called as an ampulla. So now we see there are 3 ampullae on either side. It is the sensory receptor which is now seen in these dilated ampullae and is known by the name of cristae ampullaris. So the sense organ is now called as a cristae ampullaris. Now when we go into the details of this structure, there's a sensory receptor. For an easy learning, we will consider the horizontal canal so that it will be easy to draw and learn. So let us consider this as the midline where we have the horizontal canal with the ampulla at one end, and here again we have the semicircular canal with the ampulla at one end. It is a thickening of this epithelium of the membranous labyrinth which give rise to the structure of the sensory organ. That is what is called as a crista. Together named as cristae ampullaris because it is located within the ampulla. Now this structure is actually seen like a dome-shaped structure which is a thickened epithelium of the membranous labyrinth. So there it is actually the tall columnar epithelial cells which are also ciliated. So in short, they are epithelial cells. But in between we can see there are the supporting cells also. So actually these epithelial cells which give rise to the sensory receptor are actually modified neuronal cells acting as a receptor. So they are epithelial ciliated cells, actually modified neuronal cells acting as a receptor cells. And there are in between the supporting cells or the sustentacular cells also. So these are situated within the ampulla, and you should understand there is something on top of it where there is a gelatinous structure called as a cupula. So there is a gelatinous structure overlying the cilia of the tall columnar epithelial cells which is called as a cupula. And this structure, although I have drawn like this, it will usually be filling the whole of the ampulla. So it completely obliterates itself, filling the whole of the ampulla. So this is a cristae ampullaris. The detailed structure is in such a way that we have the epithelial cells which are ciliated. On top of it there is a gelatinous material called as a cupula. And it will be totally filling the whole of the ampulla, so that whenever there is a fluid motion of the endolymph, usually it will obliterate its flow at the level of the cristae. Is it clear so far? Now, again going to the details of these cells, we should know something about this cilia. Because when we consider a single epithelial cell, which is a neuronal cell, we know there are cilia present in these epithelial cells. They are actually an extension of the cytoplasm giving rise to hair-like structures. So these hair-like structures will be in such a way that there is one tall cilium with shorter cilia directed towards one direction. This is how it looks like. So we see that there is one tallest cilia which is now called as a kinocilium, and the shorter ones which are decreasing in the height are called as a stereocilia or stercocilia. Both are the same because some textbooks say it is stereo, some say it is stercocilia. So these are the cilia which are seen, or the hair cells which are seen nearer the kinocilium, decreasing in height. But at the same time, they are connected by membranous structures which are called as tiplix. Now when you learn about the audition, there also it is the same hair cells which are acting as the receptors. So these are actually mechanoreceptors where a distortion or a displacement of the hair cells will be stimulating or will be giving rise to an action potential. So when we learn about the mechanism, we will come to know what is the stimulus for these receptors. So coming back again to the kinocilium and the stereocilia, we know that there is one tallest kinocilium. And whenever the movement of the cilia occurs, if it is towards the kinocilium, it always results in a depolarizing signal. And if this cilia are moving away from the kinocilium, it always results in a hyperpolarizing signal. So when we consider this crista ampullaris, where we have the receptor within the ampulla, there are 3 ampulla because there are 3 semicircular canals, and there are 6 total because there are left and right semicircular canals. And here, as I told you, the pairs which are working in the same plane are called as a synergistic pairs, and it will be in a pull or push fashion, and it is also a mirror image. Why I say it is a mirror image? Because when we consider the right semicircular canals with the crista or the receptor, we see that it is towards the utricle that the kinocilium will always be directed. Followed. Always the kinocilium will be directed toward the utricle. But for a better learning and understanding, you just imagine that the kinocilium will be always towards the midline. That will make learning very easy. This is because always the kinocilium, because the semicircular canals open into the utricle, the kinocilium is also towards the utricle. So when we consider 2 semicircular canals on the left and the right, the kinocilium on the right side will be just towards near the midline. Here also it will be nearer the midline. So now when we consider the neuronal signal, what will happen? If this is moving, both are moving to the left side, what will happen? On the right side it is moving towards the kinocilium, but on the left side it is moving away from the kinocilium. So the signal on the right side will be the depolarizing one, and the signal on the left side will be of a hyperpolarizing one. So the planes in which 2 canals will be working will be in such a way that the movement of the head of the body, whatever it is towards one side. So that side may be producing an excitation that will come to later, will be producing an excitatory signal. At the same time, it is an inhibitory signal on the opposite side. That's very important. This is the basis for learning how the mechanism of the semicircular canals or even the receptors of the utricle and saccule works. So basically, this is the principle behind the signal transmission from these receptors. So now we'll go to the mechanism of action where this happens. So finally, when we learn that it is the hair cells which are acting as the receptors, they are mechanoreceptors, and the distortion of these hair cells are responsible for producing a signal. Now we see that these stereocilia and the kinocilium, because of their attachment to the tip links, if it is towards the kinocilium that the movement occurs, it produces a depolarizing signal. This is because at the tip of these cilia are potassium channels. So whenever it moves towards the kinocilium, it is in such a way that these potassium channels become opened, and potassium will enter into it. Even calcium also enters the cilia, resulting in a depolarizing signal. And the innervation of all these epithelial cells will be by the fibers coming from the vestibular division of the vestibulocochlear nerve. Finally, they will be going to the vestibular ganglion, or otherwise called as a Scarpa's ganglion. That forms the first order neuron, because all sensations are through a 3-order neuronal pathway. So the first order will be the Scarpa's ganglion or the vestibular ganglion. That way it goes. We'll learn about the pathway later on. So this is how it occurs, and when there is a depolarizing signal that occurs, which is due to the change in the hair cells, when it reverts back, it finally gives rise to the closure of the potassium channels, and the depolarization signal will be stopped. Now continuing with the structure of the receptors present within the utricle and the saccule. Those are also receptors which are functionally the same. That is, the mechanism is the same, where the hair cells will be stimulated. So the structures in the utricle and the saccule will be in such a way that the direction of the hair cells are different in the utricle and the saccule. Moreover, there are also slight differences. Here again it is the epithelium that is thickened to form with in between we have the supporting cells, and these are also ciliated just like the other one. And we also have the gelatinous material, which is a cupula. But another important difference is that this cupula or the gelatinous material is studded with very many calcium carbonate crystals. So this gelatinous material on top of this hair cells of the ciliated epithelium is studded with calcium carbonate crystals. They are also called as otoliths, and hence this is otherwise called as a otolith organs. These are also called as otolith or otoconia. So this is one important difference, where this cupula or the gelatinous material is studded with calcium carbonate crystals, and so now we see that the endolymph is of high specific gravity compared to the perilymph. And because this cupula is now studded with this calcium carbonate crystals, that will add on to the specific gravity, which is almost twice that of the endolymph, and hence this is always under the influence of the gravity. So these receptors, that is the receptors present in the utricle and the saccule, are always under the influence of gravity because of this high specific gravity contributed by the calcium carbonate crystals on top of the gelatinous material or the cupula. Followed. And another important difference is that the arrangement of these receptors in the utricle and the saccule are also different. In the utricle, it is arranged in a horizontal manner, so the direction of the hair cells will be vertical. Whereas in the saccule, it is arranged in a vertical manner, so that the hair cells will be in a horizontal direction. So that is the difference here. So the direction of the hair cells will be different in the utricle and the saccule, where the hair cells are directed vertically because it is placed horizontally, and here because it is arranged vertically, hair cells will be in the vertical direction. Now we will come to the stimulus where these receptors will be functioning. When we learnt about the semicircular canals, we know that in the midline we have all the three ampulla with the cristae, isn't it? Filling the whole of the ampulla. So they are erect in the standing position of any individual. Whereas in the same way, when we consider these two, they are in these positions or oriented in these positions in the utricle and the saccule in a person who is static. But this is always under the influence of gravity. For the same reason, there will be a continuous stream of impulse that is being sent from these receptors to the brain because of the influence of the gravity, appraising the brain about the position of the body with respect to the gravity. Whereas these structures in the ampulla, they get stimulated only when there is a change in the position of the head in relation to the body. So we see that this is a tonic receptor where the otoconia are acting as tonic receptors with a continuous stream of impulses even during rest, whereas this becomes stimulated only when there is a motion that happens within the body or of the head in relation to the body. So this is acting as a phasic receptor. So these are the tonic receptors and these are the phasic receptors. But acceleration is the main force of stimulus for all these receptors. So the acceleratory force for this crista in the ampulla always will be angular or rotational. So the acceleratory forces can thus be divided into angular and rotational for the crista ampullaris, and it is the gravitational for the otoconia. And this is a tonic receptor and this is a phasic receptor. But both are stimulated by the acceleratory forces. Now when we consider the angular acceleration, when a person notes this or head by saying yes, goes in the anterior-posterior direction. So that produces an angular acceleration that will stimulate the semicircular canals or the receptors of the semicircular canals. Even if the person says no by nodding the head left to right, that is also another angular acceleration that will stimulate these receptors in the semicircular canals. Even otherwise, if the person just turns his head to the left or right, touching the shoulders, that is another form of angular acceleration that will be stimulating the receptors of the semicircular canal. Rotational, as the person spins the head, that also will give rise to the same type of angular acceleration which will stimulate the receptors of the semicircular canal. But here, the otoconia, which is always under the influence of the gravity, we are always aware when we are inside a lift. We are just aware because even if the lift is going up or down, we will be sensing it and we say the lift has started up, or we say the lift is going down, or the person as is jumping up from a platform or going up. Those all will be, the person will be knowing that when the plane is being taken off, we know it. When the plane is going to land, that also is being sensed. So all these are only because of this otoconia. So in spite of the person being stable, they are aware of the motion that happens due to the motion that happens only because the person is as a whole is being moved in a particular vehicle or whatever it is. Even otherwise, we sense it. So we become aware of these senses when there is a motion that happens. Similarly, here also we become aware of it. Otherwise, when we are just walking, sitting, lying or running, we maintain the posture and equilibrium. That's why I told you it's an unconscious sensation. Only when it becomes abnormal, we become aware of it. Just like the palpitation, we become aware of the heart. When we become dyspneic, we become aware of the respiratory system. Like that, this is unconscious sensation which become conscious when there is some abnormality and it disturbs the posture and equilibrium. Now we will come to the mechanism by which these are stimulated. So always remember, these receptors are stimulated by acceleratory forces. We will just learn how the semicircular canals are in action when there is an acceleratory force acting upon it. So here you have to understand one point where the endolymph has an inertia always. So whenever there is a rotation or movement of the head or the body, there is a movement of the endolymph and the perilymph, because the bony labyrinth also will be moving along with the motion of the head. So when there is a motion that happens in the bony labyrinth, it usually will produce a movement of the endolymph, because that forms a basis for the tilting of the hair cells. That's a basic mechanism even in audition, because there is a basilar membrane which fluctuates because of the motion of the endolymph. On top of it we have the, what's the structure in the audition? Organ of Corti. So there again it is the hair cells which are stimulated by the movements of the membrane. And again when we go to the mechanism of hearing, there are so many aspects of hearing, like the pitch discrimination, etc. Localization of sound, so many things. But basically the hair cells become displaced indirectly because of the situation on the basilar membrane. And it is the endolymph that produces the displacement. Here again, when there is an acceleratory force that comes into action, it will produce a mechanical tilting of these hair cells, and because they are neuronal cells acting as receptors, they are polarized cells, they undergo depolarization, and there is a rule whereby the movement of the hair cells, if it is towards the kinocilium, always results in a depolarizing signal, and if it is away from the kinocilium, it results in a hyperpolarizing signal. So now we learn how this comes into play. Let us consider the horizontal canals first. So we have the crista here on either side in the ampulla, isn't it? So in a person who is upright, it is erect. Let us say that this particular person is turning the head on to the right side, isn't it? When the person tilts the head to the right side, what will happen? Bony labyrinth also moves to the right side, isn't it? Now we have the cupula which was upright at rest. What will happen when the bony labyrinth moves to this side? Cupula will also be displaced to the right side. So here the cupula will be going to this side. Here again this goes to this side. That is what is going to happen. But because of the inertia, the endolymph will be lagging behind, and the movement of these cupula will be in a direction. The movement of the head will be to the right side, whereas because of the inertia of the endolymph, the displacement of the crista will be in the opposite direction to begin with. So here the head is rotated to the right side, but because of the inertia it will be standing behind, and so what happens here? This is actually, instead of going to the right side, it will be displaced in this direction, isn't it? Is it clear? So because of the inertia of the endolymph, the erect crista now will be at the initial part. Because of the inertia, they are displaced in the opposite direction of movement of the head. So when we consider the movement of the head on the right to the right side, this cupula moving to the left side results in what type of signal here? As I told you, always consider towards the utricle, you can consider it as the midline. So kinocilium is here and kinocilium is here for the left one. So here when the kinocilium is moving in the, or when the motion of the endolymph is in the opposite direction of motion of the head, here what happens to the stereocilia? Is it going towards the kinocilium, or it's towards the kinocilium on the right, resulting in a depolarizing signal on the right, whereas on the left it is going away from the kinocilium, and the left it results in a hyperpolarizing signal, isn't it? So the head is being rotated to the right side, where it results in a depolarizing signal going from the right side or right semicircular canal, but on the left side it is an inhibitory signal. But after few seconds, the momentum will be catched by the endolymph, and as in rotation continues, the inertia will be lost and the endolymph will be in the direction of motion. So the displaced crista will now come back to its original position. And when the movement continues or the rotation is continued, there is nothing happening. So there will be signals equally transmitted, or there is no stimulation that happens to the brain from either of these crista. Followed? And just when the rotation is stopped, what will happen? Again the same thing happens because of the inertia of the endolymph, and the displacement of the crista will now be in an exactly opposite way that was there in the beginning. So now here the displacement will be in this opposite direction because the displacement to the right has been stopped, so it will be again in the opposite direction, and here instead of the kinocilium, the stereocilia directed towards the kinocilium, it is going away from the kinocilium on the right side, and here it will be towards the kinocilium on the left side. So it becomes an inhibition on the right and excitation on the left at the end of the rotation. Followed? So to begin with, right side is the motion of the head. So to begin with, again it is excitatory signal that goes from the right side. The placement is, that's why I told you it's always a push-pull fashion, always a mirror image. Followed? And this is acting in the same plane, semicircular canals. If the angular rotation is to be functioned by the anterior right, the posterior left will be in the same plane. They together will be functioning. Similarly, the right posterior and the left anterior. That is why they are called as a synergistic phase. So this is what is happening, because we always will not be moving in the, if it is the same direction or vertical, means there is nothing happening. If there is a motion or movement of the head to one side, that has to be informed to the brain that the person is moving to the right side. So the signal that goes to the brain will be excitatory, or it is a depolarizing signal reaching the brain. Same time it is a left that is going, that's inhibitory. At the end of the rotation, it becomes a reverse. So both the signals are simultaneously sent to the brain, where the brain will be apprised of these signals, and the person will know that I am going either to the right or to the left. So this is the importance of this mirror image or the pull-push fashion of this functioning of these cristae in the semicircular canals or the ampulla. So when we consider the other one, there is a utricle and the saccule. As I told you, when a person moves up the lift, which will be functioning? When we are going up the lift means, what should be the direction of the hair cells? Which should be horizontal. Where is it placed horizontal? Where are the hair cells in the horizontal position? It is placed vertically in the saccule. So saccule is a one, isn't it? So if you are attentive, you will answer which is the one. If I have said it wrong, you will correct it. Utricle, it is arranged horizontally. Hair cells are vertical. So it is a horizontal movement that will be stimulating the horizontal cells. Sorry, is the utricle. And now it is a lift means the hair cell should be in the horizontal direction. So it is the saccule that will be stimulated. So never go wrong. In the utricle, it is arranged in a horizontal position, so that if it is placed horizontally, hair cells are vertical. Because if it is placed like this, hair cells are vertical. So it will be moving in this direction. When a person is moving in a car or a train. So that is why when we go in a ship, and especially if the sea is very rough, what will happen? All sorts of movements will be there. And if the person is having a very sensitive vestibular apparatus, that creates vomiting sensation and dizziness. We will come to it later on. So these movements, the vertical and the horizontal arrangement of these receptors separately in the utricle and the saccule is also with a purpose, where along with the gravitational pull by the otoconia, the person will be made aware of the motion that happens or the acceleration of the body that happens, whether it is in the horizontal direction or in a vertical direction. Similarly, these cells will be stimulated to the maximum until we attain a posture or equilibrium. When a person walks uphill, how will he walk or she walk? Uphill. What is the posture adopted when a person is going uphill? Person will be stooping forward, that is to catch the equilibrium. And here the stooping will be correspondingly equal to until the person maintains equilibrium, so far where these hair cells will also be tilted. And the tilt will be in such a way that until the person maintains equilibrium, that tilt will also be proportionately more. If a person is going downhill, he will be walking like this. This is also with a purpose, where these hair cells will be helping us to maintain the equilibrium, because they are always under the influence of gravity. That's why they are called as tonic receptors. The other one is called as the basic receptor. So now at least you have understood the mechanism, how this functions. Now we will see some of the PPT here. And this is the structure of the external ear, the external auditory meatus. Now it directly enters the cochlea. That's a portion which will be helping in the audition. Now you can also see in the same part of the temporal bone where we have the three semicircular canals, the anterior superior, posterior inferior, and the horizontal one. Okay. Now coming to the next slide, here the membranous labyrinth is being shown. You can see in the enlarged form where we have the three semicircular canals. This is a lateral semicircular, this is a posterior, this is a posterior, and this one is the anterior. And they end up in the ampulla, which is a dilated portion. These are the three dilated portions. And here it is a oval window which is related to audition, but you can see the vestibule here. At the same time, you cannot see the utricle and the saccule. Here we can see the vestibular receptors which have been shown in the form of a cut portion or a section. So we have the 3 ampullae where we have the receptors, which are the crista, and we have also receptors of the utricle and the saccule seen here in the utricle and the saccule, and the 2 divisions of the cranial nerve, the vestibular ganglion. That part which goes to the audition is the cochlear division. This is the vestibular division. This again showing both the bony labyrinth and the membranous labyrinth with all the canals. Again a cut section showing the bony labyrinth and the membranous labyrinth. So the whole structure of the internal ear is seen in this diagram. Here comes the structure of the, which is a structure, it is the otoconia or the otalis which was seen here. So it is actually the receptor seen in the utricle and the saccule. Since it is placed horizontally or vertically, the hair cells are seen in the vertical direction, so they are placed in the horizontal way, which is actually the receptor which is seen in the saccule. And if you go back, we can see these are the epithelial cells which are acting as the modified neuronal cells, which are the receptors with supporting cells in between, and you can see the cup-like structure seen on the base. They are the nerve fibers innervating these receptors, and you can see the membrane also here, the gelatinous membrane, on top of which we have the otoliths or the calcium carbonate crystals. Here again you can just see the location of the receptors in the 3 semicircular canals. These are the 3 cristae, and these 2 are the receptors in the utricle and the saccule. And this is in relationship to the position of the head. You can see this is a horizontal semicircular canal here, which is shown, and this is the right anterior, this is the right posterior. So the right anterior and the left posterior will be in the same plane, the left anterior and the right posterior in the same plane, and we have the horizontal canals or the lateral canals in the same plane. This is just to show the position of the cupula in the upright position and the displacement when there is an acceleration. So here it is in the upright position, B it is a displacement of the cupula, and when we consider this is an angular acceleration, the flow of the endolymph also showed here. So this is actually towards the utricle, where the signal transmission will be in the form of a depolarizing signal. So correspondingly the rotation and the position of the canals have been shown here. So here right posterior is the left posterior, right anterior, left anterior. So the planes will always be there, they are acting as a mirror image or push-pull action. Now it's an enlarged diagram of the crista. You can see this fully filling the ampulla, obliterating the flow of the endolymph. You can see the cilia also on top of this. Is slightly a cut section which is shown. We have the epithelial cells here, the nerve fibers, and also the gelatinous portion. This is another diagram where you can see the crista filling the whole of the ampulla, but we can also see the kinocilium and the stereocilium, where the kinocilium is the tallest one and the height of the cilia goes on increasing as we reach the kinocilium. Here you can see the stereocilia when they bend towards the kinocilium, there is a depolarizing signal. It is due to the release of an excitatory neurotransmitter which can be either aspartate or glutamate, and so the spikes will be 160 per second. It's an excitation. When it is at rest, it is only about 90 spikes. It is upright. Here again it is going, the channels get closed when the cilia move away from the kinocilium, and see the number of spikes are 20 spikes per second. It's actually the release of the neurotransmitter will be inhibited. Here this will show the cliplings. And you can see in the upright position the spiral-like structure which is a clipling, and you can also see the potassium channel. As it moves towards the kinocilium, there is a stretch of this clipling, opening of the potassium channel, wherein calcium also enters along with the potassium, giving rise to the depolarizing signal. And finally, when it gets opened, after that, the stretch on the clipling will be taken away so that it again becomes relaxed, and it gets closed. It is said that a myosin filament is related to this mechanism. Now we will see how it works. So this is the hairstyles which we have with the tallest kinocilium and the stereocilia. Nothing more is seen here, and we can see the attachment of the cilia. Here, when it is deflected to the left towards the kinocilium, there is the opening of the channels and it results in depolarization. Again it comes back, especially when the inertia of the endolymph is over and the movement of the endolymph will be in the direction of the motion, coming to the upright position. Again, when it moves in the opposite direction, it is away from the kinocilium, resulting in an inhibition. This is again another diagram of the crista, the nerve fibers, etc. Again the movement of the kinocilium and see the direction of the endolymph resulting in the movement of the cilia towards the kinocilium results in a depolarizing signal. Whereas when the endolymph is moving in the opposite direction, it results the movement of the cilia away from the kinocilium, resulting in the hyperpolarizing signal. This again is to show the position in the resting state is 90 on either side. But when there is an excitation to one side, it results in about 180, whereas in the opposite side it goes to an inhibition with 10 spikes per second. So I told you the position of the receptors in the utricle and the saccule are different, and they are tonic receptors. The utricle, where the receptor is placed in the horizontal, because it is placed in the vertical, it gets stimulated by horizontal acceleration. In the saccule, it is in the vertical acceleration because it is kept in the horizontal way. This is again another diagram showing the receptor in the utricle and the saccule. This will again show the movement of the cilia. See, head motion to one direction, whereas the movement of the cilia will be in the opposite direction. Head motion again on either opposite direction, resulting in a hyperpolarizing signal. Always the movement of the endolymph will be in the initial part opposite to that of the direction of motion of the head. So this is just to show that whenever we are having that maintenance of the posture, when a person climbs uphill, the displacement of the cilia will be to that limit until that person maintains the equilibrium. So the displacement of the cilia goes on until the person can maintain the equilibrium. So depending upon the steepness, when the person goes uphill, the person stoops forward. If the person is going downhill, he will always be leaning back. So this extent of the leaning forward or backward, as the case may be, will be depending upon the extent of the cilia also, which gets displaced until the posture and equilibrium is maintained. This is just to show the movement of the endolymph in relationship to the movement of the head. See here, this is prior to rotation. We have the semicircular canals where the cristae are upright, and so the stimulation is the same from either side, or there is no inhibition because the spike or the potentials that are being sent to the brain is equal on both sides. Now you forget about the eye movements for the time being. We'll just focus on this part. So now there is a head rotation to the left side where we see the movement of the endolymph due to the inertia, and the initial phase will be in the opposite direction to the rotation of the head. So what happens here, because of the inertia in the opposite direction, here it is towards the midline or towards the kinocilium. So it is in the direction of the rotation of the head, that means towards the left side. Here in the opposite side, what we say is it is away from the kinocilium. So there is an inhibition, whereas here there is an excitation. So at the beginning of the rotation, to which direction is the head being rotated, there will be an excitation, at the same time an inhibition in the opposite semicircular canal and the receptor, which is in the same plane. So once the rotation is continuing, the inertia will now catch momentum and the crista will come to the normal position, so it becomes upright. Again it is the same like the resting position. After that, at the stoppage of the rotation, what happens is exactly opposite to what had happened in the beginning of the rotation will happen here, where initially this was stimulated, now this is inhibited, whereas this which was inhibited will now be excited. Now this movements of the eyes will be learnt later. So now we have finished the functional anatomy of the vestibular apparatus and the detailed structure of both the type of receptors in the vestibular apparatus and moreover the mechanism of action. We will now go to the pathways as well as the function of the vestibular apparatus in the next class, and further we will learn about the abnormalities like the applied aspects and also how the vestibular function is being tested clinically. So today we will stop here. Today we will continue with the vestibular connections, its functions, dysfunctions and how it is being tested. So the vestibular function test also. So in total we are dealing with the vestibular connections first. So all this will be dealt today. So coming to the vestibular connections, this is actually a special sense which is also having the general rule where these sensations will be passed to the central nervous system. So this is also just like the 3-order neuronal pathway, and the vestibular division or the fibers of the vestibular division that supplies the semicircular organs, canals as well as the utricles and saccular maculae will be together giving rise to a ganglion which is similar to that of the dorsal root ganglion. So we have the vestibular ganglion which is otherwise also known as the Scarpa's ganglion. So we have the first order neuron which is the vestibular ganglion formed by the fibers of the vestibular division of the 8th cranial nerve, and this is actually coming from these receptors. So this forms a first order neuron, and coming to the second order neuron it is a vestibular nucleus, but here it is the vestibular nuclei because it has 4 divisions where it is the superior, inferior, medial and lateral. The lateral one is otherwise known as a Dieter's nucleus. So we have 4 groups of nuclei which together constitutes the vestibular nucleus. So this forms a second order neuron, and always it is seen that the medial one and the superior one will be always remaining together, and this is actually seen at the pontomedullary junction because the dorsal root ganglion is always outside, it enters the spinal cord. So here it enters the brainstem at the pontomedullary junction. And then coming to the third order neuron, as in other case it is the thalamus, where again it is a posterolateral nucleus as well as the posterolateral nucleus of the thalamus. But this vestibular nucleus, unlike the rest of the tracts, makes connections with very many other subcortical structures because it has a role of maintaining the posture and equilibrium. So now we will come back to this connections of the vestibular nucleus other than to the thalamus. So in total we have the tracts like the ventriculo-thalamocortical tract or fibers. Then we have coming down we have the vestibulo-ocular tract or fibers. Next is the vestibulocerebellar tract. Then the vestibulospinal tract. So these are the main connections of the vestibular nucleus. Coming to the vestibulothalamocortical tract, it's actually these fibers from the vestibular nucleus going to the posteroventral and ventrolateral nucleus of the thalamus, from where it is projected to the cortex. So which area of the cortex means it is a temporal lobe, but it is also again from the opposite thalamus, again it goes back to the opposite temporal lobe. But certain books say that it is actually being projected from the thalamus into the which lobe? Temporal. So, but it is also said that at times it is being projected into the anterior part of the primary auditory cortex. So, to be more specific in the temporal lobe, it is in the anterior part of the primary auditory cortex, because it is actually part of the division of the 8th cranial nerve which has other function of audition. So, it becomes a vestibulocochlear nerve. So, this is a coming to this, back to the thalamocortical fibers. The function of these fibers are that it is responsible for the conscious awareness of the body position as well as changes or acceleratory forces as well as equilibrium of the body, which is made conscious of by these fibers. Now coming to the vestibulocular tract, this is actually ascending up from the vestibular nuclei along the medial longitudinal bundle. So here it is the temporal lobe. Medial longitudinal bundle, and it synapses with the motor nucleus of the 3rd, 4th and 6th nuclei. That is, the vestibulocular tract rises from the vestibular nuclei and along the medial longitudinal bundle ascends up to synapse with the motor nuclei of the 3rd, 4th and 6th cranial nerves at this level. So this is responsible to give rise to a reflex called as a vestibulocular reflex. We will come to it later. It's very important. Coming to the vestibulocerebellar tract, these fibers, and before that you should understand this is to both sides. The vestibulocular fibers are going to both sides. And the vestibulocerebellar tract is also going to both sides via the inferior cerebellar peduncle to the flocculonodular lobe of the cerebellum to synapse with the fastigious nucleus. The efferent also arise back from the same nucleus to the vestibular nuclei. So we have the vestibulocerebellar tract via the inferior cerebellar peduncle to the flocculonodular lobe of the cerebellum. Again this is bilateral, which means it is going both sides. And this is responsible for the maintenance of the posture and equilibrium. As you know, this is a main function of the flocculonodular lobe of the cerebellum, again for the posture and equilibrium. Coming to the vestibulospinal tract, this is having two divisions. One is the medial and the other one is the lateral vestibulospinal tract. The medial vestibulospinal tract falls short at the level of the cervical region or the upper thoracic region, where it is responsible for controlling the muscles of the upper extremity and the neck muscles. This is short, falling short at the level of the upper thoracic region, innervating the muscles or the nuclei of those nerves which are innervating the muscles of the neck as well as the upper extremities. So those muscles are being controlled by the medial vestibulospinal tract. This is short as well as going only to one side, so it is ipsilateral. Whereas the lateral spinothalamic vestibulospinal tract is long and it falls short at the lumbar level, where it innervates those nuclei which are innervating muscles of the trunk as well as the lower extremities. So those muscles are being controlled and regulated for the maintenance of posture and equilibrium by this lateral vestibulospinal tract, and here it is bilateral. So this is long and bilateral, this is short but going only to one side. So that's the difference. So this will be regulating the muscles of the upper extremity as well as the neck muscles. Here it is regulating the muscles of the lower extremities and the trunk muscles. So that's the vestibulospinal tract. It is said that these tracts will all even otherwise making connections with the red nucleus and the reticular fibers before ending with the alpha motor neuron of these nerves supplying the muscles. It may be again either ending directly or through an interneuron. And the facilitatory and inhibitory signals coming from the higher centers will be integrated, and that is how these tracts will be regulating the muscle tone to maintain the posture and equilibrium. That is why these are having connection with both the red nucleus and the reticular fibers, because reticular fibers itself will be of two types, pontine and medullary. One is stimulatory, one is inhibitory. So the balance between the inhibitory and the facilitation will be brought about by the integration and coordination by this tract on these fibers, and that's how the tone of those muscles or the anti-gravity muscles will be made and posture also maintained. Now coming to this vestibulocular reflex. The vestibulocular reflex is a reflex whereby there will be movements of the eyeballs when there is rotation of the head. So whenever there is a rotation of the head, there will be the movements of the eyeballs in an attempt to fix the image on the center of the retina, or the images will be preserved as such in the visual field without disturbing the vision. So fixing the image at the center of the visual field whenever there is a rotation of the head or movement of the head will be initiating this reflex. So once the movement of the head is detected, it triggers this vestibulocular reflex in such a way that there will be an inhibitory signal going to the extraocular muscles of one side, whereas there will be an inhibitory signal going to the extraocular muscles of the other side. So one side will be inhibited, the other side will be excited, because already we learnt in the vestibular apparatus there is a synergistic pairs working together. Signals going to the brain will be on one side it is excitatory, the other side it will be inhibitory. So accordingly, whenever there is a movement of the head, it will be detected by the semicircular canals or the uticular and the saccular maculae, whatever it is, depending upon the type of movement that the head produces. At the same time, the signals will be going in such a way that one group of extraocular muscles on one side will be inhibited, the other group of muscles on the other side will be excited. So this is a vestibulocular reflex, and this reflex is normally present in all human beings, and if it is slightly impaired, that will produce very much distress even for reading. And the reflex can also be called as two types, where we have the rotational type of vestibulocular reflex and there is a translational type of. So there are two types of vestibulocular reflex. So whenever the movements are detected by the semicircular canals, that will trigger the rotational type of vestibulocular reflex, and whenever the movements are detected by the maculae of the utricle and the saccule, that will give rise to the translational type of vestibulocular reflex. So whatever be the movements of the head, there is a reflex movement of the eyeballs, because the head can be rotated or moved in its own axis, whether it be vertical, horizontal, rotational. So when the head is moved in any direction in its axis, the eyeballs will also be rotating in its own axis, but in an opposite direction. Followed? So the movement of the head will be associated with the movements of the eyeballs in the opposite direction, and that is a reflex movement produced by the eyeballs, and it is actually a compensatory mechanism whereby the images will be preserved in the center of the retina or in the center of the visual field. So that is the purpose of the vestibulocular reflex, which is normally present in all of us. And that finishes the vestibular connections. Now coming to the functions of the vestibular apparatus, we know there is a semicircular canals as well as the utricular and the saccular maculae. One is acting as a basic receptor, the other one is acting as a tonic receptor. So the first one, that is a semicircular canal having those receptors, will be actually helping in a what type of equilibrium? Static or dynamic? It is dynamic equilibrium. So let us imagine a person walking or running. Suddenly if somebody calls from the back and he turns ahead, or even if the person hits some obstacle, but still the person will maintain the equilibrium and will not fall off the equilibrium. This is possible because here comes the role of the flocculonodular lobe, which is an organ having a high predictive function, where it can predict the disequilibrium or the displacement that is going to happen, and it will get send the signals to the semicircular canals, where it is able to detect at the beginning as well as at the end of the movement, whatever be the type of movement that happens. It can be the angular or the rotational. So that is being detected at the beginning and at the end of this acceleration, and immediately correction will be made by signals being sent to the centers, the vestibular, cerebellar and the reticular. And so here we see that in anticipation of the displacement of the disequilibrium that is going to happen, there will be appropriate adjustments made to the anti-gravity muscles because of this particular mechanism. So all are A, in anticipation, appropriate adjustments against this acceleration, and again it is done in the anti-gravity muscles. So many A's are there. In anticipation, against the acceleration, it is the appropriate changes that happen, it is the adjustments that are made. Adjustments are made in the anti-gravity muscles. So if you remember all these 5 things, that is how the dynamic equilibrium helps us from falling off the equilibrium whenever the person is in motion and loses, and there are chances that he can lose the equilibrium and fall. So that is prevented by the semicircular canals. When it comes to the uticular and the saccular maculae, it is said that it is very much effective in its function, extremely effective, especially when it is in the near vertical position of the head. And we know the direction of these hair cells of the uticular and the saccular maculae are different. One is vertical, the other one is horizontal. So these hair cells will be stimulated in a variable fashion with the variable type of head movements. So when there is a variable stimulation for this type of hair cells to the different type of the movements of the head, the brain will be apprised of the signals going from either side. Again, they are working as synergistic phase. So here it is not in the same plane, but the same uticular or the saccular maculae will be functioning depending upon the acceleration, whether it is in the horizontal or in the vertical direction. So when the signals are being sent to the brain from either side, the brain will be appraised of the signal whereby to which side the head is being moved or the body is being moved to. So what happens is, again, this will be sending signals to the centers like the vestibular, cerebellar, reticular, etc. And again the anti-gravity muscles will be made to have to make the corrections and adjustments, and the equilibrium is maintained. So this helps in actually the static equilibrium. So it just detects the linear changes, but it cannot detect the velocity. That's why this is called as a static equilibrium. So what happens? Because it cannot detect the linear velocity, what happens? It cannot prevent the fall. It only helps in maintaining the balance. So to prevent the fall, semicircular canals will be helping the beginning of the movement and the end of the movement being detected by the semicircular canals. So that helps in the dynamic equilibrium, and this helps in the static equilibrium. That is how this function of the vestibular apparatus goes. Is it clear so far? Now coming to the vestibular dysfunction. Before that, we learned something called as nystagmus. So nystagmus is actually a proper type, another type of vestibulocular reflex. As we know, in all the normal situation, whenever there is a movement of the head, there is a movement of the eyeballs to fix the image on the retina itself, or in the visual field it has to be the proper point. So this is actually an involuntary and spontaneous movement of the eyeballs, again trying to fix the gaze when there is a movement of the head. So it is involuntary and spontaneous, not with a purpose, and this is called as nystagmus. So the nystagmus itself has 2 components. One is a quick component and another one is a slow component. To understand nystagmus further, we will just imagine somebody sitting in front of us, opposite to us, and both of us are traveling in a vehicle. Let us say we are in the direction of the movement of the vehicle in a train, and another person sitting opposite to us. We just observe that person's eyes who is looking out at the objects. So the objects are stationary, but it is moving because we are moving in the train. So this person who is focusing his gaze on a particular point or object when we are observing, we see that his eyes will be slowly moving as the vehicle is going on in a direction, and the movement will be very slow until at a point where the eyes cannot further be moved. So what happens? At that particular point, the eyes will quickly snap or jump to another point. Again the vehicle is moving, so the movement of the eyes in this particular person will be in a direction which is exactly opposite to the movement of the vehicle or the train. Again it goes to a point, reaches a limit, it again snaps or jumps to another point. So we have seen there are two components. The one which is being done as a vehicle is moving on, fixes to a point, it goes on like that and goes to one point, after that it quickly jumps. So that is a quick component. The other one is a slow component. If you observe very carefully, the eyes are actually moving, the slow component is actually moving in the direction opposite to the movement of the vehicle, isn't it? It is in a direction opposite to the movement of the vehicle, which means the person is moving, or instead of the head, now we see that the person himself is moving. The rotation of the eyes are in the direction opposite to the direction of the motion of the vehicle. And when it quickly jumps to another point, it is actually in the direction of the movement of the vehicle. If that person is looking at our eyes, again it will be the same. If we are looking out, we are looking at an object, and as the train is moving forward, we are in the direction of the vehicle, but our eyes will be moving opposite to the direction of movement. But when it jumps to another point, what happens? It is in the direction of. So always the quick component says in which direction the head is moving. Follow. So the quick component is always giving the direction of movement of the head, if we consider this nystagmus. So nystagmus is something which is not at all harmful, because it can be rotational, it can be congenital, it can be in the vertical direction, it can be a rotational type of nystagmus, anything can be there. It's only spontaneous and it's not disturbing the vision. But the visual process is not affected. Moreover, the vision is not initiating this or triggering the nystagmus. So blind people can also have this nystagmus. So it is not at all impairing the vision. And hence nystagmus at the same time is of help to find out clinically some amount of the vestibular dysfunction. That's why we are just learning about the nystagmus. And it is said that the quick component is always triggered at the brainstem level, whereas the slow component is actually triggered by the vestibular system. So that is nystagmus. And coming to the vestibular dysfunction, we have different types of dysfunction in the case of the vestibular apparatus, where the first one is labyrinthitis, which is the inflammation of the labyrinths. The second one is Menial Disease. Third one is streptomycin toxicity. Fourth is motion sickness. And this is something following labyrinthectomy. So the labyrinthitis is actually the inflammation of the labyrinth. So here what happens is the person will always be having a symptom. As I told you, whenever there is a dysfunction of the vestibular system, this is actually designed exclusively for maintaining the posture and equilibrium. So whenever there is a disturbance or a dysfunction of the vestibular apparatus, the person becomes highly immobile. It is very much distressing. And it always makes the person feel that he is going on spinning around, even though he is remaining stationary. So that is called as vertigo. So the major symptom in labyrinthine dysfunction is vertigo. And it sometimes said that it can be subjective or objective. Subjective is as a person feels himself as being rotating. Objective is He feels as if the environment is rotating. So that is objective and objective. Anyway, so that is a vertigo, and this is usually associated with nausea, vomiting and dizziness. So that is vertigo. So any person with having a slight dysfunction can have these type of symptoms. Even alcoholism can produce these type of symptoms. That is a vertigo. And motion sickness is also part of this, where the semicircular canals are highly sensitive, and so there is a rhythmic stimulation of the vestibular apparatus depending upon the type of automobile or motion that is happening because of the travel. So it can be sea sickness, air sickness, land sickness, etc. So now this is vertigo, where it is found in connection with labyrinthitis, where we have inflammation of the labyrinth. Coming to Menial disease, this is actually due to some abnormality in the endolymph. The secretion is affected, there is some more amount of the endolymph, and the condition results in what is called as endolymph hydrops. So there is excess of this fluid, especially in the endolymphatic sac also. So actually tubes of this endolymph is being kept inside for correction. And here the symptom is, or the drawback is that in spite of this vertigo, dizziness, vomiting, etc., there is a progressive deafness, especially the person becomes insensitive to low frequency sounds and it goes in for deafness. So that is a symptom here, especially for the Menial disease. And subtomycin toxicity, actually it is an anti-tubercular drug which also produces the same symptom of vertigo. It's a common complaint done by the patients having anti-tubercular treatment with subtomycin. Then motion sickness, again I told you it is a excessive stimulation of the vestibular apparatus when the person is traveling. Imagine a person who is traveling in a ship, especially if the ship is rough. What happens? There are all types of movements that are happening in the anterior, posterior, lateral as well as up and down. So all the type of movements will be happening in a travel in the ship with a rough sea. So this induces the motion sickness, and it is said that the exact mechanism is actually yet to be understood. So the person will have again all these type of symptoms. It may even be associated with hypertension, BP fall, vomiting, so many other autonomic disturbances. Now coming to labyrinthectomy, if there is a unilateral labyrinthectomy done on one side, in animals we see that the animal will be going on rolling and rolling to catch the balance and the equilibrium. But in human beings, if it is one side that is being labyrinthectomized, the person will get adjusted and adapted in 1 or 2 months. So the person can manage after that. But something which we have to understand here is that whenever a person maintains the equilibrium, there are 4 main signals that help the person to maintain the postural equilibrium. So that includes one is the vestibular, that is actually dealt with in detail in the postural equilibrium. Then we have the visual cues which help the person to maintain the equilibrium. Then we have the proprioceptive impulses. And finally the cutaneous. Let us say the person has had the labyrinthectomy, followed, especially if it is bilateral. And I told you, if the person is blind, at least 2 of these signals, if present, the person can manage. So if he is a blind person with absent labyrinth, he will manage with these 2. Follow. Now we will just imagine a person being asked to walk on a compound wall which is having a sloping top. So a normal person, as he walks on top of the compound wall, what happens? Will he be walking upright or straight? No. So how will he be balancing himself? He will balance himself with a particular posture where so many joints will be bent. Both the upper limb and the lower limbs will be in such a way that he is trying to balance himself, and there the proprioceptive impulses are there to help him to adopt that posture. And that is again helped because the platform is being sensed by the cutaneous receptors. And he can see with the visual cues where the edges of the compound wall can be looked into to protect himself or prevent himself from falling. And above all, we have the vestibular, because whenever there is a motion or a movement that happens, that will be helped by the vestibular apparatus. So now let us see when we make the person blindfold and then make him walk on the same region. So what will be guiding him now if he is made blind? He has the rest of the three, isn't it? Now we see that he is already having a labyrinthectomy and we make him blindfold. Still, can he walk? Still possible. But when these proprioceptive impulses, as we say, this is actually the impulses coming from the periphery, that is from the muscles, tendons, ligaments, etc. So the nature of those anti-gravity muscles of our body, actually we say that the flexor muscles of the upper arm and the extensors of the lower limb are actually acting as anti-gravity muscles. So the status of these muscles with respect to its tone as well as its state of contraction and relaxation of the different groups of muscles will be variable. So the proprioceptive impulses actually will be variable in the group of anti-gravity muscles to help him maintain the posture, isn't it? So he can manage somehow with the cutaneous. But let us imagine a person who is made to swim in water, in a deep water, who has had the vestibular removed. Now what will happen? Will all the rest help him? What do you say? The cutaneous can help him, but is there any platform where he can feel it? No. The proprioceptive, even maximum what he can do is only swimming. But in spite of no platform to rest or find a position where he can maintain, that's also not going to help him. And the visual cues, although not blind, under the water what can he see to guide him? So what will happen? The person will finally drown. So this is why the normal physiological question is being asked about where labyrinthectomized person should not go for deep sea diving. So that is the reason where we say these people should be careful and not go for a deep sea diving or deep water diving. So now we will go to the vestibular function test. Actually, whenever there is some abnormality in the vestibular apparatus or this sense organ, what we do is we can find out if it is normal by inducing a vestibulocular reflex in the form of the nystagmus. So it is being induced by making the person rotate in a Barney's chair. So that is a Barney's test. And the second one is a caloric stimulation test. So what else will be the significance of this test? In addition to finding out whether the labyrinths are functioning normally, we can also find out if there is a brain death. Why? If we rotate the head or move the head, we are supposed to get the vestibulocular reflex. If it is not present, that shows there is a brain death. So these days it becomes important because of the organ transplantation. So we can confirm the brain death that way also. Although that is not the final and confirmatory, we can make use of this test for confirming the brain death also. So here, in addition, clinically, even if we make the person do the Romberg's test, what is that Romberg's test? Asking the person to stand with the feet closed and eyes being closed. So will that person sway or go to fall? A normal person will not. But if there is sensory ataxia due to the lesion in the posterior column, will he be swaying to one side? Because among the four, cutaneous and proprioceptive is gone. Visual cues are gone. So naturally he will sway to one side. But this is not a test for the vestibular function. This is actually a test for sensory ataxia. But this can be positive even in cerebellar dysfunction as well as posterior column dysfunction, but not for the vestibular. So the vestibular function can be done separately in each ear by these tests. So in the Bionic test, what we do is we make the person sit in that chair and rotate the individual at the rate of 10 to 20 per second. So it's very fast. And we look for the nystagmus, the post-rotatory nystagmus. Its direction, rate and duration will be looked for. And then we compare it with the standard and say that whether the person is having some abnormality in the vestibular apparatus. But coming to this caloric stimulation test, which has to be done separately in each ear. Here what is actually done is a person is being seated, and if it is being seated, we will make the person's head being protracted backward for 60 degrees. Whereas if it is in the supine position, we make him flex for about 30 degrees. Because these uticular and the saccular maculae are very, very sensitive, that even half a degree will be detected normally in the near vertical position. Here we are just making it in this position because we make the vertical canals in the horizontal position and we do the caloric stimulation test. The principle behind this is that we will create a convection current within the endolymph by slightly changing the temperature of the fluid. So we irrigate the external ear with water which is either just above the body temperature or just below the body temperature. So we can use cold water as well as warm water. If it is a cold water that is being used on one side and is being irrigated, stimulating the vestibular apparatus or the semicircular canals, because of the change in temperature that induces stimulation, we see that the nystagmus will be in the opposite direction. So if it is a cold water, nystagmus will be in the opposite direction. And if it is a warm water, it usually produces a stimulus in the, nystagmus in the same direction. So this is the mnemonic which can be used to find out the caloric stimulation test. Whereas the same thing can have another mnemonic which is by ACTH, because away from cold, opposite. Towards the hot, same sign. So this is the code which can be used to conclude the test, whether that particular part or the side of the vestibular apparatus is normal or not. If it is abnormal, we cannot perform the test. There will be no response at all. So that's how we conclude. We can also compare with the standard. But all these are become very obsolete because of the development technology. So these tests are only based on these principles where we induce the vestibulocular reflex in the form of nystagmus, looking for its rate, direction and duration. Because the post-rotated nystagmus will usually be there for 30 to 60 seconds. By that time, we have to detect the nystagmus and then report by comparing with the normal in the case of this Bionic test. Here, it is actually we are looking at the direction, everything based on the type of temperature. So here, cold will be usually done first because that is acting as an inhibitory stimulus, whereas warmth will be acting as an excitatory stimulus. So that is how the vestibular function test can be studied, and technology will show very many newer techniques for detecting the dysfunction or abnormality in the vestibular apparatus. So that we finishes the vestibular function test also. Thank you.